All right, so I think we'll get started. Well, thanks everyone for coming to Annie's defense. I remember when Annie first walked into my office and she started talking about bridge flank hydrothermal systems, which at that point I had no idea what they were. And then she started to talk about, she started to talk about how she had published a paper on octopuses. I thought Hannah was coming. and on DT octopuses. And I also don't know that much about DT octopuses, but Annie's enthusiasm, perseverance, and goal-oriented personality made me realize that she would succeed in whatever she was doing. And luckily, I persuaded her to study a little bit more about benthic communities, which is something I do know about. And so Annie received her PhD, her BS, not yet, in geology right here at UNH. Then went on to a master's in oceanography at URI. And in between her master's and PhD, she worked as a research associate and research technician at Bigelow Laboratory in Maine, and also at the University of Alaska in Fairbanks. While doing this, she had the opportunity to participate on multiple research cruises and even dove in a submersible, and you can see that, you can see her pictured next to one, which I honestly am in awe about, because I would never ever do that. And while pursuing her PhD, she took advantage really of all that UNH has to offer. She completed certificates in ocean mapping, geospatial science, and a cognate in teaching, in college teaching. Since starting her PhD, she has gone on numerous cruises and presented at many conferences. And I could really keep going, but I'm sure we all want to kind of hear her talk about bridge flank hydrothermal systems and the communities associated to them. Before I do, I just want to kind of acknowledge her committee members: Michael Pallas, Sema Dykstra, Richard Smith, and Giuseppe Massetti, who is online.
Yes, I am.
Okay, great, good. nice to hear you, Giuseppe. And with that, I'll give the floor to Annie.
Thank you, Jen. Hi, thank you all for coming to my dissertation presentation. So like Jen said, I'm going to be talking to you guys about community composition and environmental variables on ridge flank hydrothermal systems. And I do this to explore relationships between the environment and life in the deep sea and apply it to habitat suitability models, as well as the impacts of fine-scale bathymetric resolution on seafloor characterization and habitat suitability models. And these are pictures of me in the submarine at 3,000 meters of water and then coming out of it. And it was a very special dive because it's on the sites, one of the sites that I'm presenting on today, and it really inspired a lot of my research like in the past decade. So what you're seeing on the screen right now is warm fluid venting from the deep sea floor. And that's the shimmering or fluid seeing. That's where the warm water is mixing with the cold bottom water. You're also seeing a couple octopuses, shrimps, and then a temperature logger. And you're looking at a ridge flank hydrothermal system. A ridge flank hydrothermal system is a section of the deep sea floor where fluid is entering some recharge outcrop, flowing through the crustal aquifer, becoming heated up, and then it discharges at a discharge outcrop. The fluid that's coming out of the seafloor is elevated in temperature relative to background. So if the background water is about 2 degrees C, the fluid coming out of the seafloor is measured at 12 degrees C. And it's depleted in dissolved oxygen by about half. However, there are no other significant chemical differences between the fluid that's venting out of the seafloor and the ambient bottom water that it's venting into, at least that are measured. As such, these sites are really middle ground environments between the cold deep sea and really extreme hot vents that people are more familiar with. The cold deep sea is a relatively homogeneous environment. The community that lives there is cosmopolitan, so it's widely distributed. Its distribution is related a lot to the type of seafloor that it can attach to, as well as food availability. And on the other end of the spectrum, hydrothermal vents are very extreme environments The temperatures that are venting at these sites are typically hundreds of degrees C warmer than background. They're significantly chemically altered, and the communities that live there are very much endemic. So there's a specific community that's occurring with a specific vent zone, and oftentimes they're reliant on symbionts. Ridge-flank hydrothermal systems have this warming, but it's absence of the chemical alteration, and so they're really ideal sites to be able to study the impact of warming on community compositions in the deep sea. This can be important because we know that in the deep sea, in the Pacific Ocean between 1 and 4 kilometers, that warming has been occurring at rates of 1/100th to 1/10 of a degree per decade, and it's expected to occur for decades into the future. This is related to climate change and slowing of ocean circulation. And ridge-flank hydrothermal systems are especially helpful for trying to understand the potential impacts of warming related to climate change, because once the fluid discharges from the outcrop, it very quickly mixes with that cold background water, and then the temperature anomalies measured around those vent sites are on the same order of magnitude as those that we measure from warming related to climate change, and we expect to continue into the future. So they're about 10ths to 100ths of a degree elevated above background. It's important to understand how warming could influence deep sea communities because most of life on Earth is expected to live in the deep sea or near the bottom. We've only mapped about 26.1% of the ocean floor at high resolution as of June 2024, and we've explored even less. And when I say exploration, I'm really talking about using underwater vehicles that are equipped with cameras and probes and samplers that kind of give us an opportunity to get our eyes and our hands on the deep sea so that we can try to understand relationships between the environment and the types of organisms that live there. Those relationships can be really important because they help us inform models that allow us to predict species distribution elsewhere. So we call these habitat suitability models, and therefore predicting species distribution. And what they do is they take known occurrences of different species or families on the seafloor, and then they pair it with predictor variables such as temperature or the shape of the seafloor, and then they output predicted distribution of those different families based on the probability that different environments will be suitable for that family to occur in. They're assessed by how accurately they're able to classify both the test samples, so the occurrence data that we're putting into it, as well as the background data in which those animals are occurring. So those are the AUC and the gain. And just as a note, in my dissertation, I create my habitat suitability models with a program called Maxent. One of the issues with habitat suitability models is that they are impacted, the species distribution that they are predicting is impacted by the resolution of the data that is going into it. So this can be a particular issue for seafloor variables, such as the shape of the seafloor or the texture of the seafloor. We know that in shallower regions, so the continental slopes, that a grid difference between 1 kilometer resolution versus a couple hundred meters resolution is going to impact how the models are predicting species distribution. But it's less clear about how differences in fine scale bathymetric data, so between like 1 and 10 meters, are going to impact species distribution models in the deep sea. As technology is advancing and we're able to equip underwater vehicles with high resolution, high frequency multi-beam systems, we can collect very, very good data on the deep sea, but it's unclear how differences in fine scale resolution is going to impact those models. So that's one of my questions. So in this dissertation, I use ridge-flank hydrothermal systems to study the fine scale links between the community composition and the environmental variables. And I apply those to addressing 3 questions or 3 chapters. In my first chapter, I look at the relationship between biodiversity and temperature. In my second chapter, I'm focusing on how fine scale resolution is going to impact habitat suitability models as well as seafloor characterization. And in my third chapter, I actually apply some of those relationships to habitat suitability models for a number of families on one of my study sites. So this research uses 2 different study sites, the Davidson Seamount Management Zone, the Monterey Bay National Marine Sanctuary, which I will refer to as DSMZ for the rest of this talk, contains 2 of the discharge outcrops, the Octopus Garden and the Octocone, and then the Dorado outcrop, which is off the coast of Costa Rica and where I was fortunate enough to do the alpine dive. So like Jen referenced, both of these sites have very well documented aggregations of brooding octopuses. So they are special for octopuses, and we know that. But there's a whole community composition that really hasn't been studied before this dissertation was completed. All three of the outcrops are less than a square kilometer in size. At max height, they're only about 150 meters, and they're all in about 3,000 meters of water depth. So they're very similar environmentally. The Dorado outcrop was explored, or the data that I'm using from the Dorado outcrop is from 2013, and it was explored with ROV Jason. Jason was equipped with a camera, so we get identification of the seafloor and what's on it as it flows through the water column. And then it was mapped with AUV Sentry, also in 2013. And AUV stands for underwater vehicle, ROV stands for remotely operated vehicle. They're both robots you can put down on the seafloor. And Sentry also had a temperature probe, and so it was, as it was mapping the seafloor, it was also collecting data on what the temperature of the water column was. Davidson Seamount Management Zone was explored in 2019 with ROV Hercules, and Hercules was equipped with both with cameras as well as the temperature probe, and then it was mapped in 2021 with AUV Dorado, not to be confused with the Dorado outcrop, they're different things. So from the visual observations that the ROVs collected, I was able to identify both the substrate types, so whether the bottom was hard or soft, as well as the families, the megafauna families that were in the dive footage, as well as their locations. I did this with the aid of different identification guides from University of Hawaii, Mambari, and NOAA, as well as the help volunteer hours of an undergrad named Anna Wheat. And from all of the observational temperature and mapping data, I was able to create a fine-scale habitat map to help me address my questions. Before I jump into each individual chapter, I do just want to explain how I am using the bathymetric data to extract environmental variables related to the shape of the seafloor and the texture of the seafloor. So, very crude explanation of how we collect depth data about the seafloor. From either a ship or some kind of underwater vehicle, or surface vehicle, we can attach multi-beam systems. They send sound out, it bounces off the seafloor and returns back to the ship or the vehicle. Depending on how long it takes for that sound to travel to the bottom and back, as well as the sound speed through the water column, we're able to get estimates of depth. We can grid different compilation of estimated depths and create a digital terrain model. And from that digital terrain model, we can extract information about the shape of the seafloor, what I'll be referring to as geomorphons. So if you think about this like the slopes and the ridges and the flat areas, as well as information about the texture of the seafloor or the terrain. So this is like the roughness or the peakedness, and specific variables that I'm using are northernness, easternness, depth, standard deviation, slope, and ARB-DMV, which I will explain in a later slide. So for the classifications of the geomorphons, I use a program developed here at CCOM, and it was described by Massetti in 2018, and it uses a radial search pattern to classify each center node. So within a group of nodes, the center one is classified based on the ratio of how many of the surrounding nodes are higher or lower than it. There's user inputs for this, including the search radius, as well as what's designated to be a flat. And then based on that ratio, it's going to classify each center node or each area as a different geomorphon, so slope, a ridge, and then there's an option of 10. For the terrain variables, I use a tool within ArcMap called the TASI Toolbox. This was described in 2015 by LaCourse, and the TASI Toolbox uses a 3x3 search window to define a number of different variables. Here I'm using 6 of them, and together they're supposed to explain 70% of the variation within the seafloor, as well as have limited collinearity between them. So for depth and standard deviation, which is the standard deviation of the depth value, the algorithm uses all of the cells within the 3x3 window. For the RDMB, which is similar to the bathymetric or topographic position index, and stands for the relative, is also referring to the relative difference to the mean value. It's looking at the entire local mean within the window, subtracting the focal mean, so the center node, and then dividing that by the range within that local mean. And then for the northernness, easternness, and slope, the algorithm uses Horn's method, which gives doubly weight to the north, south, or east-west direction, depending on if it's northernness and easternness. Those are both aspect or the directionality of the slope values. So now I'm going to jump right into my first chapter, where where I'm focusing on the relationship between community composition and temperature in the deep sea. And I do this by comparing community composition in and outside of vent zones and the correlating environmental variables. So I use a series of analysis to compare my community compositions between the vent zones and the non-vent zones. Just going to quickly brush through these, and then I will be presenting the results as I go through this presentation. So to determine how different environmentally my vent zones and my non-vent zones were at each site, I use the t-test just to compare average environmental variables. To establish if I've sampled enough families to actually do analyses that can inform about community composition, I'm using a species accumulation curve. To determine if my vent sites, so the community compositions within my vent and my non-vent zones, are significantly different and how different they are, I use multi-regression permutation procedure as well as a cluster analysis for visual representation. I can determine how consistently different families are occurring within each zone type using the indicator species analysis, as well as determine my diversity indices within each vent zone type, or within each zone type, and then compare those against the environmental variables using the least squared regression. And finally, I can determine how environmental variables are correlated with community composition within each specific zone type using a best analyses. And I do this chapter, I focus on both the Dorado outcrop and the DSMZ, so both of the outcrops. So the vent zones were initially selected based on temperature data at Dorado outcrop, where zone sites were selected based on areas around active venting sites that had temperatures anomalies that were measured to be at least 1/100th over the average ambient background temperature. I also had 2 vent zones that are relatively close in proximity. I really wanted to treat them as separate analysis sites, and so that limited the size of the vent zones. For the non-vent zones, I selected those to maximize the similarity between the environmental variables of the vent zones with the non-vent zones. So what I really wanted to be doing was comparing areas with different temperatures, but that were otherwise the same. And so I ended up with 4 vent zones and 4 non-vent zones, all with 34-meter radiuses. For the Davidson Seamount Management Zone, I applied the same approach. I'm going to point out here that there are 2 discharge outcrops within the DSMZ, but for this chapter, I've treated them as if they're one site, so the Davidson Seamount Management Zone as a whole. And so the zones and the vent zones are distributed between both the octopus garden and the octocone. And applying the same method, I get 4 vent zones and then the 4 non-vent zones. So comparing my environmental variables, I can see that temperature is significantly different between vent zones and non-vent zones at both Davidson Seamount as well as Dorado. And that geomorphons are also significantly different between vent zones and non-vent at Dorado. And northernness is significantly different between vent zones and non-vent zones at the Davidson Seamount Management Zone. This information I used to inform which environmental variables I then was going to use to determine which environmental variables were correlating with community composition and the best analyses, as well as applied to the least squared regression when I look at the relationship between diversity and environmental variables. The establishment of if I have enough samples. So this plot is a family accumulation plot, and it's what I use to determine if I've sampled enough to be able to do a community analyses. And you're seeing plateauing lines for both vent zones and non-vent zones at both of my sites. And because they plateau, that is an indicator that with all of the images that I looked at along the dive track, I was able to observe enough families that I am getting a relatively solid sample of the community, so I can be doing analyses that may inform about the community. From my cluster analysis, visually, I can see that there is a separation between the vent zones in orange and the non-vent zones in gray. This is true at both Dorado and the Davidson Seamount Management Zone. They're a lot more, the separation is a lot clearer in the Davidson Seamount Management Zone. And then assessing that with the statistical analysis with the MRPP, I find that the vent zones and the non-vent zones at both of the study sites are significantly different. The MRPP also can report how different community compositions are. So while they are significantly different, there might be some overlap between which families and how many are occurring in each zone type. And that's indicated by the t-value. The lower t-value at Dorado suggests that there's more similarity between the 2 different community compositions compared relative to the high t-value at Davidson Seamount Management Zone, which is indicated in the higher t-value. And you can see it in the cluster analysis. It's a lot sharper separation. From this indicator species analysis and looking at how consistently a family is occurring within a specific zone type, I find that the Davidson seamount values, so a higher indicator value, the IV score is representing more consistency, are almost twice as much at Davidson seamount than they are at Dorado. And this aligns with the finding that there's a greater t-value, more separation between the community compositions between the different zone sites that I observed with MRPP. I will point out that brooding octopuses is an indicator species, family, or genus. These aren't identified to the species level. For both sites, and that's not surprising as the brooding octopuses are clustering around these venting sites, and we know there's an association there. And that glass sponges are an indicator value of the vent zones at Dorado, which those are really good nutrient cyclers and could be important for just ecosystem dynamics. For my non-vent zones, the indicator species analysis found a lot more variety of all the different species that indicate each zone. Again, at Davidson Seamount, the indicator values are a lot higher than at Dorado. This again lines with that higher t-value and more separation between the community composition. Interestingly, the Brazingid sea star is an indicator species of non-vent zones at Dorado, but vent zones at Davidson Seamount Management Zone. This may be related to their ecosystem function within each of those groups. It could be other external forcings that are just controlling their distribution, but they are tolerable to the temperature range that we're observing on the outcrop. And I'm going to point out that the squat lobster is an indicator species for both the non-vent zones at Dorado and the Davidson Seamount Management Zone. So when I look at the diversity indices, I only found significant differences between richness and Shannon diversity on the Davidson Seamount Management Zone within the vent zones and the non-vent zones. And this isn't necessarily surprising. The Davidson Seamount Management Zone contains the Davidson Seamount itself, which is a lot higher. Seamount is over 1,000 meters tall. It has a large, diverse community composition that could be supplying warm or temperature tolerant larval and organisms to the two octopus, so the two outcrops, the octopus garden and the octopone that could be establishing. Whereas the Dorado is a lot more isolated in the Pacific Ocean. Off Costa Rica, there hasn't been a major seamount that's yet been identified in that area that would be supplying that same type of larval supply. The least regression squared analysis, which compares the diversity indices, so just the richness and the Shannon diversity, against the log of the temperature, found that there is a significant correlation at both of my sites between richness and Shannon diversity and temperature, and that at Davidson Seamount, based on the R-squared adjusted value, I can see that the deviation in those diversity indices is explained almost twice as much by temperature at Davidson Seamount relative to at Dorado. This could be related to the fact that there is this warm fluid is venting from the site. If the fluid is greater in volume at the Davidson Seamount Management Zone, it could be making a stronger temperature impact, driving a greater temperature differential. However, more sampling as well as exploration is needed to determine if that hypothesis is correct or not. And finally, for the environmental variables that correlate with the community composition within each vent zone type on Dorado and Davidson Seamount, I find that on Dorado, temperature correlates with community composition in the vent zones, geomorphons in the non-vent zones. And then at the Davidson Seamount, temperature and northernness both correlate with community composition within the non-vent zones. So from the statistic analyses that I ran and the comparisons, I am confident to say that temperature does correlate with community composition and diversity. It is impacting it. In the North Atlantic, a similar relationship was found between temperature and richness, and the authors explained that this was related to perhaps a larval supply that was tolerant to these warm temperatures. Now, when I first introduced this dissertation, I talked about climate change and how ridge-like hydrothermal systems may be good for assessing climate change. There is a caveat to extrapolating our found relationship between temperature and community composition to climate change, because climate change has a lot of different impacts on the deep sea. such as on dissolved oxygen, as well as food availability. And those are both, for example, aspects or factors that could be influencing community composition and diversity and need to be considered when discussing climate change. But our results do indicate that temperature is playing a role in diversity. Another important takeaway from this chapter is that the ridge-flanked hydrothermal systems are very similar seemingly environmentally, right? They're this low temperature, what one might describe as boring fluid discharge. But the community compositions are quite different. While there is some overlap, they seem to have their own dynamic systems, and as such, each ridge-flanked hydrothermal system should be studied for itself, even though they're small and deep. And these results came out, I published them in the spring in a Nature portfolio, Communications Biology. In my second chapter, I focus on fine-scale resolution impacts on seafloor characterization, as well as habitat suitability models. And I do this by regridding a 1 meter bathymetric grid at 2, 5, and 10 meters. And then I do a number of analyses across those resolutions. I look at how resolution is impacting characterization of different seafloor shapes, so the geomorphons, by different seafloor terrain, so like the texture. I look at how richness changes across resolution for specific geomorphons, as well as how the correlation between those environmental variables and community composition change across resolution. And finally, I look at how resolution is impacting the output of habitat suitability models for 2 different genuses, or genera, the black coral, as well as the sea lily. Now, I do this analysis on just the octopus garden and the octocone. This is because that bathymetric data was collected in 2021 by a group at Bombari. It's very well cleaned, it's very well documented, and I trust the highest resolution data set in regridding it to 10 meters. So when I talk about differences in resolution between 1 and 10 meters, the left panel is showing 1 meter grids underlain by either a 1 by 1, a 2 by 2, a 5 by 5, or a 10 by 10 meter box. When I resample it, all of the grid cells within that black box is being averaged and accumulated to give me the resample value at the 10 meter, 5 meter, 2 meter, and I didn't need to resample the 1 meter resolution. And then to extract the terrain variables, that's using the 3 by 3 window. And so for each of those grid cells, or the grid resolutions for the 10 meter grid, all of that information within the gray box is being used to calculate the quantity of the value of the terrain variable in each individual grid cell. And kind of the takeaway from this slide is that the 1 meter gray box is a lot smaller than the 10 meter gray box. So there's a lot more information that gets incorporated into the calculation of the terrain variables at 10 meter as opposed to at the 1 meter. So what I'm showing you on the screen right now is 6 different terrain variables that I focused on. Down looking at across resolution, so 1, 2, 5, and 10 meters as you get up to the bottom of the screen. And not unexpectedly, each of these different terrain variables had a characteristic response to the differences in resolution. So for the standard deviation, this is the standard deviation of the depth, you can see a general brightening of the image as you go down screen. And I will point out also that below each figure is the range of values depicted in each picture. And so for standard deviation, when you're looking at the discrepancies of depth, as you increase your area, especially on a sloped outcrop, you would expect you're going to have more variation in your depth. And so this is a surprise. Similarly to slope, slope is calculated based on the change in elevation over a distance. With these outcrops, they are quite sloped, and as you zoom in, you can see a lot of different variation in places that are steep and shallow. And so as you expand your area, you might be incorporating more shallow values into your slope calculation, which would explain why it's shallower, why the slope shallows with the lower meter resolutions. For the RDMV value, you can see that the boldness of the feature actually increases with the lower resolution. Now, RDMV is equivalent to the bathymetric position index, or the peakedness. And so if you're looking in little grid cells, then the peak within each little grid cell is going to seem like a significant peak. But as you expand your grid cells, the actual peaks in that larger area are going to start to stick out. So you would expect a boldening effect. And then for northernness and easternness, I don't see a range change between resolutions. And this isn't unexpected because these are both based on directionality of the slope, and the change in resolution isn't necessarily going to impact what directions your slopes are facing. So these are the same results with the same characteristic responses on octocone. For the octopus garden, for the extraction of the geomorphons, it was a little more difficult because there's a little subjectivity. As I explained earlier, there are user inputs with respect to the search radius as well as the flatness angle that go into the extraction of geomorphons. The goal was to extract the identical position and size of geomorphons at each of the resolutions, so 1, 2, 5, and 10. The reality of that was actually quite hard because there's a balance between extracting artifacts from the grids and also avoiding extracting holes or blank areas in the grids that aren't classified. It did take a bit of trial and error, and I settled on these 5 different extraction methods settings. So the inner and outer radius are written below each image. But to get kind of a feel of how well I was able to match the size or the area taken up by each different geomorphon type, I compared the areas. And so that's the bar chart on the bottom. Slope is dominant on these features. It's just so big that it's hard to see how the area changes across resolution. So I'm going to take away slope. And you can see that for the smaller geomorphons, there is variation in the area covered. It matches better between the 5 and 10 meter resolution relative to the 1 and 2 meter resolution for some geomorphons. It's really quite variable. And then the same thing for the octocone. Again, slope is such a substantial part of these geomorphons. These outcrops that I take away that geomorphon, and then you can see the variation in the area covered for the smaller geomorphon types. So to characterize my geomorphons based on my terrain, I use a program called QGIS and a tool within it called the raster statistics for polygons. And what that does is for each geomorphon, you can think of the geomorphons are broken into different subfeatures. And each subfeature is a cluster of cells where the cells are touching by 2 or more nodes. Within each cell for a subfeature, it has a value assigned for the different terrain variables, so slope, northerness, easternness, ergosity, or RDMB, standard deviation, and depth. And then the raster statistics or polygons tool takes the information for each subfeature and determines the average value, the min, the max, the range, a number of statistical suites for each subfeature. And then with that information, I compared the average value of all the subfeatures within a geomorphon type across the resolutions using an ANOVA. And I used the ANOVA to determine if, just looking at the average value of how geomorphons were characterized by each different terrain, did it change across the different resolutions? And I didn't find a specific pattern on what geomorphon or what Tasio variable changed the most. I found a mixed bag. When the ANOVAs were significant, which suggests that there is some significant difference between the characterization at some level, then I ran paired t-tests to try to find which 2 or which pair of resolution was significantly different consistency, if that was even the case. And so I'm showing you the percent of the results of t-tests that were significant. So it's just a way to say, hey, like half of the tests I ran between all the different types, all the different resolution pairs were either significant or not significant. And what I found is that in general, less than 50% of any t-test ran were significant, except for standard deviation. Standard deviation for both the octopus garden and the octopus cone, and the octocone, were significantly different for all pairs of resolutions, 86 and 96% of the time. And this is reflected when I look at the box and whisker plots to actually display the range and the average value of each of my terrain variables across each of my on each of my different geomorphons across my different resolutions. I continue to see this pattern for the standard deviation where you have an increase in both the average as well as the range. And again, that's not unexpected because when we look at how the terrain responds, the standard deviation responds to those lower resolutions, get a wider range, you're looking at a greater depth discrepancy over those slopes. For looking at how resolution impacted the richness across the different geomorphons, I did this for a handful of geomorphons. I focused on convex slope, shoulder, and regular slope. Convex slope and shoulder are both smaller areas compared to slope. And so I was not surprised to see that there's greater variation in the richness across the different resolutions on the smaller geomorphons relative to the larger geomorphon. This is because while the extraction does shift between the different resolutions, slope is so large, it doesn't really shift that much. So both the position and the area covered aren't really changing that much between the extraction levels compared to the convex slope and the convex, or in the shoulders. And what this means is that, I'm looking at community composition on a track line, and the position of that track line relative to the smaller geomorphons could just be shifted. The representation of how those positions could be shifted at the different resolutions. So that would explain why you'd see different richnesses. For the correlation between the community composition and the environmental variables, I just focused on the largest geomorphon on slopes. I find that there was no consistency between if there was significant correlations or what environmental variables were significantly correlated. And this was not necessarily surprising, as while the response of the terrain variables is characteristics with resolution, the kind of position of the highs and lows might shift a little, as you can see in those images. And so, again, the relative position of what's represented to be the occurrence of a community member or a different community composition, and the value of the terrain at the pixel in which it's occurring at would shift between resolutions. And so it's not necessarily surprising that I'm not seeing this consistent correlation. And for the habitat suitability models, for both the black coral as well as the sea lily, I did find that resolution changes the output, so the predicted distribution that's output by the habitat suitability models, but it's not impacting it in a consistent way. So you're not always necessarily predicting less or more highly suitable habitat at the lower or higher resolution. It might, in this example, the 10-meter resolution of highest probability, so these are probability bins, probability of suitability is greater than 0.5, 0.7, and 0.85. Between, at the highest end of probability, so greater than 0.85, 2, 5, and 10 are pretty similar. They are greater than 1. And then similar for the sea lily, there is variation, but there's no consistent variation. I will point out that for the habitat suitability models, I'm going to be presenting a number of results for the rest of this talk. I've reported both the gain value and the AUC, which, just as a reminder, those are how well the background grids are identified as being suitable, as well as how likely the present samples are predicted correctly. So the takeaways from my second chapter are that resolution is impacting terrain and geomorphons. And this is having an impact on why we're seeing no patterns to characterization, as well as why there's differences in community composition with the resolution. And then the terrain variable response could be explaining why there's differences in the correlation between the community composition and the environmental variables, as well as why we're seeing a lack of pattern between the habitat suitability model predictions. The implication of these results is that just because you're working at fine resolution, so between 1 and 10 meters, if you're working at 10-meter resolution, it may not be worse than working at 1-meter resolution, depending on your survey goals. The bathymetric resolution at the finest scales didn't necessarily uniformly constrain the habitat suitability model, so it wasn't necessarily improving it in any specific direction. This can be really important guiding information, because a lot of surveys may cover an area that is in over a depth variation that could cause a shift in your resolution within a survey, as well as situations where you have a sonar configuration or obliquity angle or integration errors that might be impacting the resolution of your data, or if you're using one or more data sets to do an analysis in an area. The results of this talk, of this chapter, really emphasize the importance of reporting your methodology for characterization, as it has a huge impact on how your seafloor is represented at the different resolutions. And then in alignment with chapter 1, this chapter demonstrates that the ridge flake hydrothermal systems, while similar environmentally, are actually quite different in how they're responding and characterized. So they should be studied for what they are as individuals. Does anybody have a tissue? I'm sorry. Oh, hold on, thanks, Dad. Thanks. For my third chapter, I've created habitat suitability models for 7 families and 1 genus at a range of temperature conditions. And I did this for ambient background, as well as 100th, 1/10, and 0.5 above ambient background temperatures for 7 families and 1 genus. I focused this chapter just on Dorado. Dorado, both outcrops, or both areas were explored with ROVs, but the survey in 2013 at Dorado was a bit of a grid pattern. It was designed to cover a wide area of the outcrop and get a good overlook of what everything was, whereas at Davidson Seamount, it was much more just about getting there and trying to see what we could find. We were looking for octopuses. And so it's not as uniformly covered. And so this chapter just focuses on Dorado. The families that I focus on include 2 coral families, as well as 3 sponge families. And sessile means that they are anchored to the seafloor, so they're not moving. And then I also focus on 3 different mobile families, so families that can move. Brazilian sea star, a squat lobster, and then this isn't a family, this is the genus. It's the moose octopus. It's the octopus that occurs on the outcrops and is associated with these ridge flake hydrothermal systems. I'm going to point out here that I only focused on octopuses that were not brooding. And this is because the brooding octopuses are so clustered around the venting sites, it actually makes the occurrence data, so it's less than the recommended sample size for applying these habitat suitability models. And so I'm only presenting results for non-brooding octopuses. This is the distribution of these different families in the genus around the outcrop. You can see that each little black point is representing the occurrence data. There's a lot of them and they're pretty well distributed. The predictor variables that I use in these models include temperature, and again, there's 4 different grids of temperature. There's ambient, 100th, 1/10, and 1/2 of degree above ambient. Substrate, both temperature and substrate, the original data is along the track line, and so I used an interpolation method to extract that to the entire grid area that I wanted to be modeling in. And then I also used geomorphons, depth, standard deviation, RDMB, northernness, and easternness. So I'm going to go through the results of these different families. And then all the result pages are set up similar, so you have a picture of the family, and then across the top of the screen is the 4 different grids, each representing the change in suitable area, where red is highly more suitable than blue. On the bottom is the bar chart that shows the difference in area of bin probability, so greater than 0.54, greater than 0.7, and then greater than 0.85. And on the right is the percent contribution of the different environmental variables for each of these models. And then under the picture is both the AUC and the gain value. I forgot to mention that an AUC value that's greater than 0.85 is generally considered to be reliable or helpful. And the higher the gain value suggests better predictability of each models. So for all the models within my dissertation, I had gain values that were greater than 0.8, most of them greater than 0.85, or AUC values greater than 0.8, and gain values that were usually greater than 1. So most of my models are pretty reliable. For both the coral species, I found an increase in predicted distribution with temperature, with the greatest increase between 1/10 and 1/100th, and that the strongest contributing environmental variables to the model were geomorphon, depth, and substrate, and this is true for both of my coral families that I looked at. Again, the greatest jump is between 1/100th and 1/10 of a degree, and geomorphon, depth, and substrate were the greatest contributing variables. For 2 of my 3 sponge species families, I found that, again, there's an expansion of predicted distribution with warmer temperatures, with the greatest jump for Euliptilida between 100th and 1/10 of a degree, and that depth and substrate are the greatest contributing variables. Faraday, the second glass sponge, didn't show an increase beyond the 1/100th of a degree temperature increase. Depth and substrate are the greatest, stronger contributors to the habitat suitability models here, but substrate is at 50%, and so that may be why we don't see an expansion with temperature. And for the last glass sponge, I also find an expansion of distribution. Again, there's a large jump between 1/100th and 1/10, and then also between 1/10 and half a degree. And depth and substrate are the greatest contributors to these models. So one of the important implications of seeing a distribution increase of both the corals as well as the glass sponges is that these are both foundational species that are important for biodiversity as well as sustaining a healthy environment. So their distribution in warmer temperatures could imply that there may be a boost in biodiversity if the deep ocean was to see warming temperatures, or when the deep ocean sees warming temperatures. For my squat lobster, I found that there really wasn't much expansion with warmer temperatures. This wasn't necessarily surprising when I think about in chapter 1 how the squat lobster was an indicator species for my non-vent zones. So we know that they're typically associated with those colder values, and geomorphon's depth and substrate were their greatest contributing factor. For the Brazingate sea star, similar to the squat lobster, I don't really see much expansion. I do see a little expansion between ambient and 1/100th, which is not unexpected, as this was an indicator species for vent zones at Davidson Sea Mountain, non-vent zones at Dorado, so we know that they can tolerate that full temperature range. One of the implications for seeing the expansion of foundational species such as corals and glass sponges, and a non-expansion or lack of expansion for mobile consumers, is that while we could see a boost in biodiversity in warming oceans, we would expect that the community composition would look different than what we see at Dorado, because some of the organisms at Dorado or families at Dorado aren't going to expand as well. And so you'd expect other mobile consumers could come in and fill those niche spaces. And so the community dynamics could be different than what we're seeing at Dorado. And finally, for the octopuses, the non-brooding octopuses, I find a pretty substantial increase between the 1/10 and half a degree increase above ambient background. Temperature was also a large contributor to the distribution of these models. This wasn't surprising. The non-brooding octopuses, while they often moved in and out of the zones, they were often observed around the brooding octopuses. Now, this expansion has to be kind of considered in the reality of what these octopuses are doing on the ridge-like hydrothermal sites. They are using the sites as reproductive hotspots. So their attraction to the site might be because that's where the brooding octopuses are. And so even if the ocean warms and they could tolerate it, they might not move. They might just stay where they are. So kind of the takeaways from this chapter are that there is the potential for expansion of foundational species under warming conditions, which has the possibility of increasing or boosting biodiversity in the deep sea. And this aligns with my findings from chapter 1. Again, one of the caveats to making direct statements about the definite expansion of communities and biodiversity in a warming ocean is that there's a lot of external forces related to warming that I described earlier, as well as the fact that habitat suitability models really just assume everything always stays the same forever, and it doesn't take into account for species interactions or how species interactions could change over time. So there are limitations to the interpretation of the results. An important takeaway is that depth and substrate were often and always strong contributing variables to the distribution of these organisms. At this site, that's not necessarily surprising, as most of the community is occurring on the shallow, hard substrate of the outcrop itself. This would be where you would expect more food available from currents, as well as more food available from mobile consumers. Depth is known to play an important role in the distribution of organisms in the deep sea. Usually we observe its impact over hundreds of meters, but our findings suggest that perhaps depth is actually also playing an important role on much smaller outcrops in the deep sea. And these findings are important because they can help inform for future planning, protection, and management of the Dorado outcrop. So I've demonstrated to you, hopefully, how I use fine scale links between benthic megafauna and community and environmental variables in the deep sea, and found that temperature does contribute to community composition and biodiversity. Resolution impacts seafloor characterization and habitat suitability models, but not in any kind of uniform or predictable way. And that temperature can drive an increase in family distributions of foundational species. Again, the ocean is a very dynamic place. There's a lot of factors. It's three-dimensional. Each of the factors has its own individual impact on each of the families and how their physiology responds. And so it's hard to make blanket statements about how the ocean is going to change. And ongoing exploration is definitely, sampling is definitely imperative to try to untangle some of these community and ecosystem dynamics at each of the individual sites. But from this dissertation, I can confidently say that biodiversity in the deep sea could increase with warming temperatures, and that habitat suitability models may not be improved with the highest resolution. And this can be really important because if you're working in the deep sea, you're probably on limited resources, whether it's time or money or equipment. So if you can accomplish your goals with surveying at a 10-meter resolution over a larger area or in lesser time, compared to a 1-meter resolution over a smaller area, which is going to take even more or the same amount of time, you might choose to use your lower resolution if you're going to be able to achieve the same goals. So I'd like to acknowledge the Tycho Fellowship, which is my funding source, as well as the NOAA grant here at CCOM, as well as the Earth Science Department for taking me on as a doctoral student. I'm very thankful to my advisor, Jen, for her support and kindness to me as a scientist and as a person. And my committee members, you have a proposal committee, a dissertation committee, and then a comprehensive exam committee. So they do give a lot of time, and it's very much appreciated. And then Sema Dijkstra actually stepped in on my dissertation committee last minute, as one of my members couldn't be here. Dr. Tom Littman, he answered a lot of questions as my graduate coordinator. I'm very appreciative to the admin staff, both in CCOM as well as Sue Clark in Earth Science, and the outreach staff, as well as the maintenance staff, as well as the IT department, because there's so many problems that I didn't have to deal with because all those other people took care of it for me. And that was beautiful. I'm very thankful to my professors and all the courses I've taken, as well as in CCOM or science through the cognate of teaching and learning and the geospatial science certificate, because I could expand what I was learning way beyond oceanography. And to the late Karen Von Dam, who was my professor here as an undergrad for oceanography and totally switched and encouraged and inspired me to switch my trajectory from engineering to earth sciences. Former and current lab members and peers at CCOM and NOE and Earth Science for their friendship and their misery and their camaraderie, as well as interactions I've had because of the relationship with the graduate school, the Research and Communications Academy, and the School of Marine Science and Ocean Engineering. I'm thankful to scientists that are external to UNH that have facilitated my participation in the exploration and research of both Dorado Outcrop and the Davidson Seamount Management Zone, as well as to Lindsay G. and Aaron Heffron and the Ocean Exploration Trust for opportunities to sail and fill some of the gaps in our ocean maps that aren't necessarily related to my research, but I think are very important to do. And then to my friends from home and past educations, and of course to my family, to my parents, my grandparents, my brothers, and John and Hannah, and to my cats. And with that, I'd like to thank all of you for being here, and I'll take any questions.
So we have time for questions. What we'll do though is we'll have everybody but the committee members ask questions, and then if there's if there's a few more minutes, then the community members can ask some questions, but otherwise we'll do that after.
Jack? Will?
Thank you for a great talk. That was super interesting, and I love all this stuff. So I was really interested in the second chapter. Did you do any correlation of the geomorphologic variables to something that might be a little bit more biologically focused, so like exposure for filter feeders or availability to hide for your octopi or something like that? Was that something that you took into account?
That was not. The second chapter, it's a good idea, but I very much focused on just how resolution was impacting kind of the sea floor terrain, because what I didn't really get into is in the deep sea, right, we only have 26.1% mapped, and that's not that much. But from what we have mapped and then explored with visual observations, we can try to understand, okay, so this organism is occurring on this type of sea floor. And then we can hopefully use those informations to inform where we might find other families like that, to inform where we could explore more. And so I was just interested, and this is very CCOM kind of motivated, is how the bathymetry resolution is impacting those relationships. And so biology got kind of. Any other questions? Kim?
Could you go to slide 23? No, sorry, 21. My bad.
It's okay, I'd much rather this one.
When I look at those two clustering, on the right, with all this kind of clustering, you can go as far down in the tree as you want. On the right, all you really need is one split, and you've got the two. You've got everything correct. And on the left, you've got to get down. You never actually get to a point where you get a perfect split, but the left branch is pretty good. Now, ecologically, does that imply on the right? That sort of implies on the right, in those conditions, that you've got a below or above, and that's all that matters. If you get that much above, you can't have the organism, but as long as you stay below some threshold, you can. The one on the left doesn't suggest that ecologically. Is that just a function of the data, or is that actually a reasonable environmental supposition that on the right, the conditions that you're in, the temperature does matter very much, whereas on the left it does not?
I don't suspect it's related to the specific environmental conditions. I think it's more related to perhaps the organic carbon content that's available in the vent zones that Davidson came out. There was a paper that came out in 2023 that looked that suggested that the high organic carbon content within vent zones will support is supporting the octopuses that are there, which in theory could support more organisms, whether from their senescing bodies as well as their egg casings might be attracting organisms. So the community dynamics at Davidson Seamount, I think, are driving why we're seeing differences in how we're interpreting the separation between the vent zones and the non-vent zones at the two sites.
Okay.
Does that answer your question?
Yeah, absolutely.
Val, I want to give actually a special shout out to Val. When I was very neophyte here, I came to his office with like this data file XYZs, and I was like, what do I do with these? Like, how do I get these into a map? And he took the time to show me in MB systems and extract them, and I was able to do a lot of research with that alone. So thank you for that.
My question is about like the multi-resolution study, and I guess my question is, I'm not sure how to articulate it. I'm not entirely sure I understand the details of what you've done, but to what extent do you think your results are kind of a function of the math more than the underlying distribution of critters? That is, like, you, as you were explaining them, you frequently had like a difference as a function of resolution, but you're like, but that's because like you were able to explain it away quickly because it was covering a larger area, or you would, it's sloped, and you would imagine that if you do that analysis over a bigger range, you get a bigger difference in depth or something like that. And I guess what I'm getting at is like, how do you tease out the things that are just like the math and versus like what's meaningful and useful for prediction?
I think that the math has a huge role in it. I'm only using one type of algorithm to extract these terrain variables, but there's a lot of different other options I could have used. I think to get at how the math is contributing to why we're seeing differences in the resolution, you would take an approach of extracting a suite of terrain variables across different techniques, and then comparing how each of those terrain variables based on the different techniques changes along one resolution, and then also you could expand that to multiple resolutions.
Okay.
Maybe I didn't understand your question, if you wanna.
No, I'm not sure how to best ask a follow-up question. Let me think about it.
Any other questions? Juliet?
Great job. Yay.
Thanks.
So how much do you think, kind of building on your question, if you have super high resolution data, whether that's 2 centimeters or a meter or 10 meters, how much do we not know about the ecology of how these critters live in these environments versus like, we know it's a large scale structure and that's great versus this is, can I characterize that this is a super craggy porous thing that's gonna attract different organisms that way? Like, is that something that you think that should be a variable that you're tracking?
If we have that information, I think it would be a great variable to include in analyses like this. For Dorado, there was no sampling done of the families, which is one of the reasons I only identified down to the family level. As of 2013, they actually went back last year and did a bunch of sampling at the site. And so we're just beginning to learn about the physiology of these organisms that could help make clear or reveal why, more specific reasons of why they're actually occurring in that specific spot, whether it's related to the porosity of a specific organism that they're occurring on or the seafloor. But that was one of the driving motivations behind the kind of asking these questions and figuring out how to address it was, if we're assuming that an organism is occurring on slopes on the deep sea, and then we're using slopes as our predictor variable, that might be incorrect if the organism is actually occurring on slopes because of the roughness of the slope or the smoothness of the slope. And I was curious if, by looking at how the texture of the slope is changing across resolution, is there a difference? Like, to get at, is slope an appropriate proxy if it doesn't matter between 1 and 10 meter resolutions, how rough it is, it's always gonna be represented the same roughness. So yes, I think knowing more about the physiology of why these organisms are occurring would be very important, and also that's a good question because I guess that's kind of the original question that I had when I designed this chapter. I don't know, did I answer that?
I have another question.
We'll let Will go. Like ping pong.
Yeah. This is also somewhat related to her question as well, and just kind of curiosity on my part, but in the coastal ocean, for a long time, a lot of communities were assumed to be very cosmopolitan, and once genetic analyses started to come about, people began to realize that there's actually a lot of genetic distance between these communities that we thought were the same. So I guess you mentioned that a lot of deep sea communities were very cosmopolitan. Do we know, like, how cosmopolitan are these vent communities? Are they the same critters between these vents in the Eastern Pacific, or are they different? Like, what's going on with these communities between the two different sites that you looked at?
That's a good question. One, I should kind of go back and say, from what we've explored, they seem to be cosmopolitan in the deep sea, but we have a lot of exploration there. So I found that I was able to use the same identification guides, which were based on analyses in the Pacific Ocean, to identify all of the families, I would say 90% of the families that I observed in my dive footage. Some of the families couldn't be identified just because it wasn't in the guide, or it just wasn't a clear image, but it obviously was something different than what else I was seeing. And so I'm reasonably comfortable saying that the types of organisms that we see, at least the morphotypes from just an observational point of view, do seem to be occurring. They can occur at both of the bridge-link hydrothermal system sites we looked at, as well as other places based on the guides I used. So I would say that they are cosmopolitan. Also, an analysis looking at the microbial community at the site in 2014 by Mike Lee revealed that the microbes that are occurring are not those that we associate with vent communities. And so it suggests that there's no kind of, when I think like hot vent communities, not these kinds of communities, which suggests that there's really no symbiotic relationship between the megafauna as well as the microbes at these sites. So they're not like, I would say they're more likely cosmopolitan than they are endemic.
Thank you.
You're welcome. Juliet,
So one thing that I know a lot of oceanographers, even physical oceanographers and geological oceanographers, they don't mention all the currents in the deep sea and the interaction with the seafloor, like Diane left there, but like that those Bernoulli effects, that's scale of things that people here study in coastal zone and a lot of those things are happening in the deep sea where there are still super fast currents in the deepest part of the ocean, and people didn't expect that. Is anyone measuring anything besides you're at about what's going on with currents nearby?
as far as I know, with like currents flowing over the outcrop, I'm not sure if there's active studies on that. I mean, I know that there's a lot of people looking at the currents coming out of the outcrop, but I do agree with you that because of the bottom currents, because they're going to be accelerated over those features, and the hard substrate, it's hard to tease out which one is a stronger contributing factor, but those are likely playing a role on why we're seeing the majority of these occurrences on the ridge part of the outcrop, outside of those vent zones. So yes, I think it plays a role. I don't know if anybody's doing specific studies on it. Gabby?
Yes, wonderful job. Really great talk. The research is so cool and interesting. On a personal note, now that this chapter of your life is over, what's next?
It's funny, it's not over yet. I still have to publish 2 of these chapters. Well, I am actually gonna just focus on my family for a little while. And then hopefully, well, I do plan to stay in sciences. I'd like to continue to contribute, whether through government, industry, teaching. I'm pretty open. Kim and I were talking about how it's more about problem solving than necessarily oceanography. I do find the ocean very interesting and a very happy place. So it would be a privilege to stay in the field. But for now, I'm 8 months pregnant today, so let's do this and then figure out what's next. Any other questions? All right, thank you all so much. Oh wait, Will, are there questions online?
I did not see any. I solicited the audience, but we can solicit them audibly one more time and wait 60 seconds. If no one chimes in, then we can call it.
There are donut holes and clementines on the table. If anybody wants them on their way out. All right, that's it.